Hi everyone, my name is April Zhang, and today I will be talking about my senior research project fellowship through the T-BioInfo organization. So my project is called Predicting COVID-19 Patient Severity with Machine Learning. So here is a brief overview of the topics that I will be covering today. I'll touch on what inspired my project to the methodologies, and then finally wrap up with the results of my project. And with that, let's jump right in. So last March, our world changed in ways that I couldn't even imagine. From the masks we're wearing right now to curfews, lockdowns, practically everyone has been affected by COVID-19 in some way, shape, or form. There is about 3.24 million deaths and 155 million cases, and these two numbers are constantly growing. Last summer, my mom actually contracted COVID-19, but fortunately she only had mild symptoms and recovered quickly. Of course, I was incredibly grateful, but extremely curious as to what exactly distinguished my mom from the millions of hospitalized patients that were struggling for each breath that I saw on TV. So it was this experience that really prompted me to pursue this topic as a senior research project. Okay, so these probably look very familiar to you guys, right? Everybody sitting here probably, has done temperature checks or been asked if they experience these symptoms. But what differentiates these common symptoms of mild cases versus life-threatening lung injury that many severe COVID-19 patients face? So again, here you can see SARS-CoV-2 has a few stages depending on the disease severity. About 80% of patients stay within the mild to moderate illness, which you can see is that light blue curve, whereas 20% of patients progress onto severe illness. This is where ventilators are required, or hospitalization, and even acute respiratory distress syndrome. So again, here you can see, I know this diagram is a little bit complicated, but if we just focus on these little pink alveoli, so the alveoli are the gas exchange sacs of the lungs. Here you can see the difference between healthy, infected, moderate, and severe. So there's a buildup of fluid within both the moderate and severe cases, and this is what causes the gas exchange to become more difficult, hence shortness of breath. Okay, so now that we know what the virus causes between mild and severe cases, let's look at host immune response. Host immune response is really important to look at because how the immune response reacts, how the immune system reacts, will ultimately determine disease severity. And there are, of course, many factors that contribute to a patient's reaction. So in severe patients, typically a cytokine storm is common. This causes a widespread and detrimental impact on multiple organs. Lots of severe patients are also characterized by a hyperinflammation signature, high levels of neutrophils, which are a type of white blood cell that releases cytokines, which are responsible for the inflammatory response. Okay, so having established that, my project objective is to identify and investigate what genes that are responsible for the different disease severities. So put simply, I want to explore the correlations between gene expression and disease severity, whether that's a mild or severe COVID-19 disease case. And I did this via machine learning. So the type of data that I chose to analyze is RNA sequencing data. So using RNA-seq, we can identify what proteins are being transcribed, which tells us which genes are being expressed, upregulated or downregulated. And using this information, I can investigate the relationships between gene expression and disease severity. So the output of RNA sequencing is a gene expression table. The data I downloaded from online was a gigantic gene expression table with over 19,000 rows, genes which were included. And then here you can see this image describes the basic RNA-seq workflow, and for my project, I've worked mostly with the bioinformatics portion. Okay, so I obtained my data from NCBI's publicly accessible database. One of my data selection criteria was to have a data set with more than 100 samples. This is really important for machine learning, mostly because the algorithm has more examples to build a model off of, which will improve the model accuracy. And the data that I ended up choosing was in transcripts per million and was derived from tissue origin of leukocytes, which is just the overarching term for all white blood cells. Here you can see this is an image of some of the possible data sets that I was investigating, and I organized them by number of samples and other clinical data that the researchers may have provided, for example, age, sex, etc. Okay, so after the data is collected, it's really important to normalize it, as many machine learning algorithms assume a normally distributed data. So to do this, I performed quantile normalization with a threshold of 5. I also performed a logarithmic transformation and eliminated any low expression data, which I did via Python. So here, this is just a code block of some Python that I wrote. This was for transposing data files. So I actually originally tried to do this with Excel, but the program could not handle a table with over 19,000 rows, so I ended up having to do that in Python. And then I also did eliminate any low expression data. For example, any genes that have 0 expression in over half the samples, those genes were eliminated. Okay, so the next step is the machine learning portion, which is kind of the bulkier part of the project. So before I jump into this, what is machine learning? Machine learning is a type of AI that takes input data and investigates the different patterns between groups within a training data set, and then will apply what it's learned to a test data set. And this type of machine learning is known as supervised machine learning. So this diagram is actually really deceptive because there are many intricate parts to machine learning, for instance, optimizing your parameters. And before doing this project, I had never heard of these algorithms before, right? Random forest, SVM, and naive Bayes. So with no previous experience, this was one of the challenges that I had to overcome throughout this project. And then I accessed all of these pipelines, all these machine learning algorithms via the tBioInfo platform, where they have it all nicely set up. Okay. So the final step of my project is to put what I'd found in a biological context. So using David, which is an annotation tool, I was able to functionally annotate the 14 genes that my algorithm had selected. And I'll talk a little bit about how I got these 14 genes later. I also conducted a thorough literature review, investigating the role of a few of these 14 genes in COVID-19 based on previous findings. Okay, so here is a table of the different machine learning algorithm trials that I ran. So you can see they're grouped by mean decreased accuracy cutoff, abbreviated as MDA, the training model, and then the test data classification, training model and test data classification accuracy, So mean decreased accuracy basically represents how important a gene is to accurately classifying a set of data. So for instance, if I were to take out a gene that had a higher positive mean decrease accuracy, for instance, 3.7, then the model accuracy would decrease by that number, 3.7%. Whereas if I were to remove any data that had negative mean decrease accuracies, that would improve the classification accuracy of the algorithm. And so I focused on this 1.7 mean decrease accuracy cutoff with the SVM algorithm because this trial had a reasonable balance between the training model accuracy, which was 95.12%, and also minimum number of features. And usually with machine learning, we want to identify the minimum number of gene features that can still accurately classify between groups. In this case, mild versus severe. So this is why I ended up using this trial run. So out of the genes that I'd selected, I investigated 2 of them and found them to be linked in disease severity, and I'll go more in depth for a few of these genes in the next slides as well. But here you can see this is just a brief snapshot of all the 14 genes that I found. Okay, so the first gene I investigated was CCR5, which is a chemokine receptor protein that's located on the surface of white blood cells. So previous findings have shown that the CCR5 expression is significantly upregulated in SARS-CoV-19 infected cells. So if we were to put it in the opposite way, that means if we inhibited this gene expression in severe COVID-19 patients, this would help to reduce inflammatory cytokines and decrease the SARS-CoV-2 RNA in plasma. So in simple terms, identifying this gene and its role in aiding COVID-19 infection, this allows us to intervene and possibly inhibit CCR5 expression and help control disease severity. So this could be a possible therapeutic target to help treat severe infections. Okay, so the next gene that I looked at was H6PD, which is an enzyme that's really important to reducing oxidative stress and preventing cell destruction. So past findings have shown that this G6PD deficiency affects about 10% of African American males. So again, in simple terms, having a G6PD enzyme deficiency may put African American populations at a higher risk for developing severe symptoms due to COVID-19. And of course, this may be a plausible explanation as to why African American populations seem to be harder hit by COVID-19, with higher death rates 3 to 6 times higher compared to other races. So you're probably wondering, why is this research important? This is useful because in the future we can do a simple patient blood draw and then run an RNA-seq analysis to see what genes are upregulated or downregulated. And then, for instance, some of the genes I discussed before could possibly serve as predictors for severe or mild COVID-19 disease severity. So in turn, this can really help doctors know what to expect and more effectively treat patients in the future. And then with that, I'd like to give a big thank you to my on-site advisors, Mr. Cioni, Ms. Starenfield, and then my off-site advisor, Dr. Kaur, for their invaluable mentorship and guidance throughout my research project.